So tonight's video is going to focus on evidence of evolution and things that can help support the theory of evolution and how there's a wide variety of these things. So evolution is supported by pieces of evidence from a variety of disciplines, including things like geographical evidence. So by geographical evidence, we mean evidence on location, like you can see in this bottom picture here on the left. If you look at the fossils, you can also see how there is some geographic evidence by seeing them in all these different areas that you wouldn't expect. Geological evidence would be referring to rocks and fossils. So this is what most people think of when they think of evidence of evolution, right? They think fossils, they think dinosaurs, okay? And they provide, rocks and fossils give information. Unfortunately, it's incomplete information, and we'll talk a little bit more about that in a minute. Another way that that evolution can be supported with evidence is physical, or you can maybe think of it as structural evidence. Things like homologous structures, vestigial structures. Again, we're going to talk about each one of these in detail as we move further through. Okay? We also have biochemical evidence. So biochemical evidence would be like amino acid sequencing from your freshman year when you would compare the amino acids to one another. Looking at so that would also allow you to compare the DNA as well as the final proteins that are made from those amino acids. So that biochemical support for evolution is one of the strongest supports. Okay? And then we also have mathematical data can help support the theory of evolution. Okay? Allele frequencies in particular. Okay? Think back to our last topic that we did, right? It was Hardy-Weinberg. And remember, Hardy-Weinberg was a way to quantify if evolution was happening. If those allele frequencies changed, then that gave us evidence that evolution or change was happening within the population. So mathematical data can support evolution as well. So let's start with the fossil record. So the fossil record is, unfortunately, like we said in the first slide, it is considered incomplete. Things are washed away, the rock is broken down, things are lost. Okay? Maybe only part of the animal was, covered and trapped. So it is incomplete. However, as a general rule, it can still show us patterns of evolution because we can usually see the passage of time, variety of species. So it'll show patterns of evolution, which will allow us to make comparisons between both extinct and extant species. Extant species would be current species that still exist. On the graph over here on the right, you can see this progression with horses. We can compare these extinct species of horses to the current horse species, and we can see the patterns and the change throughout. Okay? It also allows us to help document the formation of a new species. Sometimes that can be a little bit more difficult when we have the gaps because, again, the record is incomplete. And we can date those fossils, and by dating those fossils, that can help us, again, see the patterns, make the comparisons, start to date when the species were changing, what was happening there. So we can use a variety of methods to help date the fossils. We can look at the age of the rock surrounding the fossil. If you remember, the fossil is formed, right, by the silt. Will cover the silt or the sand, the dirt will cover the bones of the organism or cover the organism. Okay? And then eventually over time, between pressure and more, dirt covering, it will eventually solidify. More so than just the regular bone. The rate, you can look at the rate of decay of isotopes, including carbon-14. Okay, so basically we can look at the carbon breakdown within the fossil, within the rock. We can also use geographical data to help us date the fossils. Okay, you can see, for instance, in this picture here, you can see all these different layers of rock. Okay, and all of those different layers of rock, we could, if the surrounding rock has a certain date that we can pinpoint, that would also allow us to pinpoint the fossil age. structural evidence is another way to help support the theory of evolution. So one of the first pieces, or one of the main pieces here, is homologous structures. If you remember from your freshman year prefixes and stuff, homo means same, right? So these are structures that have similar characteristics. Basically, they are similar in structure, yet different in function. Okay, and again, you saw examples of these when you were in ninth grade biology, right? So similar in structure, different in function. You can see that here on the left, okay, with the cat and the whale and the bat and the human arm here. The same basic structure, right? The two what are called long bones, and then a series of short bones followed by long bones again. Okay, but very different functions, right? We're talking about the flipper of a whale, the wing of a bat, a human arm, a cat leg. Another structural piece of evidence would be vestigial structures. So vestigial structures are structures that are no longer functioning. So these are a non-functioning structure located on the organism, in the current organism, and it was once important to the organism. So in its history, it was once important to the organism, and the structure becomes less necessary due to natural selection. So let's look at the example here for whale evolution, right? So if y'all remember, whales, the thought is that whales actually evolved from land mammals, in particular a wolf-like land mammal. And so whales have what we call a vestigial pelvis. You can see it right here. Our pelvis is part of what allows us to walk upright, to distribute the weight so that we can walk upright. So marine animals don't need a pelvis like we have because they're supported by the water all the time. Okay, and so this would be an example of a vestigial structure. It is significantly smaller in size because that usually will happen. It will shrink over time because the structure is no longer functioning. They don't need it in the current organism. However, in the ancestral organisms had it at one point in time. It was a working, functioning structure. So both of these things, homologous structures as well as vestigial structures, show links back to common ancestry, right? These structures are going to be determined by DNA, right? The blueprints, which means there has to be some common ancestry DNA-wise in these structures here that are all coming out looking or appearing the same. On that note, then, we can talk about our biomolecular evidence, our DNA and our protein sequencing evidence, right? This is all going to go together, right? Remember our central dogma here of DNA to the mRNA to the protein. And remember that our monomer of proteins are our amino acids. So the more closely related an organism is, okay, the more DNA bases and therefore the more amino acids the organisms are going to have in common. And so you can, excuse me, you can compare the genes for the proteins from different organisms. One of the things you have to be careful with is that obviously you have to compare the same protein. So you have to use proteins that the organisms have in common. So you can't compare genes for protein A in one organism and protein B in another. It has to be from protein A in both of them. And we can use those sequence comparisons to show common ancestry, to build cladograms, to show evidence of evolution. Okay? If we look at the diagram that we have here, right, we have the human. Okay? And then you can see they've counted the differences here between the various organisms. And so the human and the lamprey would be the least related because they have the most differences. Okay? The fewer differences, the more closely related. And again, this is one of the pieces of evidence that many people give a lot of credence to. They put a lot of support behind this, and they feel very strongly about it, about its scientific, it's the quality of this scientific evidence. So we'll do some comparisons with these. We'll practice some comparisons again like you did in ninth grade. We'll use these comparisons you'll be able to build cladograms and phylogenetic trees using these comparisons. Hopefully we'll be able to run some software that'll allow us to compare, in real life, various organisms to one another. The last one we're going to look at here are molecular and cellular features. right? So these again will go along with our DNA in our protein sequencing, right? Molecular and cellular features, it tells you here they are conserved among species that share ancestry. What we mean by conserved, meaning that they're still going to exist, they're going to be retained in species that are going to share common ancestry. Okay, genes, structures, functions, they're going to keep, we're not going to start over. The organism is not going to start over. They're going to keep reusing them. Okay, things like the genetic code. If it's already in the DNA and it works, there is no reason to try to start it over. The process of photosynthesis, right? The process of photosynthesis, of protein synthesis, right? The process of that DNA to mRNA to the protein. Okay, on that note, though, the process of photosynthesis, cellular respiration, those are conserved processes. Those are processes that are similar in a variety of different types of organisms, as well as particular proteins. Usually, when we compare proteins, okay, we do look at hemoglobin and cytochrome C. These are proteins that are common in a wide variety of organisms, and so you can compare those two. You can compare organisms with those. Okay, if we look at the graph that we have over here on the right, because again, this has been a very data-heavy unit, so you're going to keep seeing more and more graphs. You'll notice this graph has its error bars on it, right? You can see your error bars forming right there. Okay, quick reminder on error bars, right? If they don't overlap, right? They don't overlap, then we would say there is statistical significance, right? If they do overlap, then we can't say that there's a statistical significance or there is no difference, right? This is helping capture our true mean or true proportion, depending upon what we're looking at. We'll look at some more, we'll review error bars with some more data in class. But if we look at this graph, it's looking at the percent of amino acid change, okay, on our y-axis there. And then it is looking at millions of years since they diverged from one another on the x-axis there. Okay, so if we are looking here, you can see how cytochrome C has millions of years since divergence is getting bigger way over here. Right, so like plants branched off a long time ago, and then what we have up here are a bunch of different kinds of animals. Okay, you can see that our percent of amino acid change for cytochrome C isn't very big. So we should see a lot of similar amino acid sequencing and DNA sequencing to build that particular protein. And so we would then be able, looking at differences there, we would then be able to say, okay, this is more related to this, which is more related to this, but this one is the farthest because it has the most percent change, right? You can see the plant out here with the highest percent change. So again, we'll be looking at a lab that will help us hopefully compare some real life organisms to one another. So again, same as always with this unit, we'll be looking at a lot of data regarding practicing with data and FRQs, open-ended things with this material.